Okay, before we get into 3.5, let's do some review. So, hypothesize what you would observe if you forgot the counterstaining step when looking at a gram-negative bacterial cell. So, the cells would appear dark pink, the cells would appear purple, the cells would be colorless and difficult to distinguish, the cells would wash off the slide. So think about what the counterstaining did. If you need to flip back to the procedure, do that and pause the video. Think about that. Okay, the answer would be the cells would be colorless and difficult to distinguish. So if we remember the order for the gram negatives, they're purple. We apply the alcohol decolorizing, and that washes out that purple stain for the negatives. They're clear at that point. And then we apply our counter stain, the safranin, which is supposed to turn them pink. But if we forget that, they would stay colorless. So C is our answer here. Hint, hint, this one might show up in many places on a test, maybe even in the lab test. Okay, just quickly, we're going to talk about some different types of microscopy. This section in the book has all kinds of cool info, but it's way beyond our needs. So if you're like super into microscopes like me, it's a great read, but I'm not going to put this on your homework and I won't test you on it. Except fluorescent microscopy, which is important here because it's super useful in clinical diagnosis. All right, let's do a case history here. We have Jared, previously healthy 48-year-old man, submitted to the hospital after 6 days of feeling weak, short of breath. When he went to the hospital, he has other symptoms: muscle pain, which we call myalgia, nausea, vomiting, and diarrhea developed there. So still pretty general. By the time he's admitted, he's feverish, flushed, and he's got this dry cough going on. So we're going to do a chest x-ray, and we're going to look for pneumonia, which we see some evidence of there. Remember back to that first tuberculosis x-ray where there's wisps in the lung x-ray. We can see this mass of cells starting to appear in the lower left lung. So before this happened, Jared had cleaned out the filter of an outdoor hot tub. And so we're going to take cultures from his sputum, that's the phlegm that comes up, and we're also going to go sample the hot tub filter and grow these on plates. And when we get them, we do the gram stain on them. That's the first thing we always do on bacteria. And we find that they are gram-negative bacilli. Now, to you, that doesn't say a lot, but to a clinician, based on where it's found, what it's doing, and the gram stain, they instantly suspect a bacteria that you may have never heard of called Legionella pneumophila. So if you've ever heard of Legionnaire's disease, this is the bacteria that causes it. And it likes warm, wet places and can spread through the air from that warm, wet water. It's named Legionella because the first outbreak that it was identified in was actually at a group of American Legionnaires that were all gathered at one hotel for a meeting, and this was being distributed by the air conditioners from that warm water from them. And so they called it Legionnaire's disease after that when they identified the causative bacteria about it. Very interesting story about modern kind of microbial detective work there. Luckily, we suspect this, and we have a fluorescent antibody that is very specific to just this bacteria. So we take a slide of the bacteria and we apply this antibody. If it sticks, then it means the bacteria will glow, and we can see glowing bacteria here because the antibody is stuck to them. That tells us for sure that it's Legionella pneumophila because these antibodies are super specific. So that tells us everything we need to know. We can treat with IV amoxicillin and clarithromycin antibiotics for 8 days, and he makes a full recovery. All of these steps in identifying it tell us the proper antibiotics to prescribe, because some antibiotics will kill some bacteria, others will kill others. They don't always work cross, so we need to know what's in there. Okay, this was all possible because of fluorescent microscopy. And fluorescence is a special type of light. When we shine a light on something, if it gives off light at a specific wavelength, we call that fluorescing. For our purposes, just think about it as glowing. So what happens here? Well, we make an antibody, and antibodies are these little Y-shaped molecules that are part of our immune system, and they target very specific things called antigens, which are little molecular shapes. They recognize them very, very specifically. So this cell here is a bacteria, and on its surface, it has little molecules that our antibody recognizes and binds to. If this is Legionella in our case, it has the specific antigen that our antibody binds to. We apply it, it sticks to it, and then we wash the slide, washing off any unbound antibody. And then we've attached to it this glowing little green molecule. And when we excite it with the proper wavelength of light, it glows green. And this is really great because we don't have any background stuff. We only see this green light. So we only see positive signal. There's no like dust or background images, things like that. And this is really powerful because not only can we detect specific microbes, we can use a stain that actually can tell if they're alive or dead. These red ones here are dead. They glow red. They fluoresce red. These live ones are green. They fluoresce green. Or we could even stain individual little parts of cells. So this is a gut, some gut cells here. And inside of it, we have all these Vibrio cholera, which are bacteria that are fluorescently stained green and are glowing green here. So we can see inside of a human cell that it's infected with these Vibrio cholera, which causes the disease, cholera. So fluorescence microscopy is really, really super powerful. We could even see individual parts of a cell if we had the proper labels for them. Now, I just want to finish to reiterate that we've talked about light microscopes here, right? Even our fluorescent microscope, it's a modified type of light microscope. That has a lower end resolution, right, of like, 0.2 is where we can kind of stop seeing, 0.2 micrometers. Viruses are much smaller than that. This is 50 nanometers, so 0.05 micrometers. So these are HIV viruses, virions, as they're called. They're budding off of a human T cell here. And here is our SARS-CoV-2 virus that we've all been dealing with. And on the outside are these little spike proteins. These spike proteins, our immune system recognizes them, and that's actually what our vaccine trains our immune system to recognize. So our vaccine trains our antibodies to recognize these spike proteins and attack the COVID virus the instant it gets into your body, not allowing it to replicate. So these are electron micrographs. So remember, we can't see viruses. They're too small. We need electron microscopes, which have a higher resolution because the wavelength of an electron is smaller than the wavelength of light. All right, that does it for chapter 3, and that is all for our first unit. So you've made it. Make sure you're using the study guide and study for that first test. If you have any questions, send me an email. All right, bye for now.